Good afternoon, everyone. In the last class, we are actually in the lesson of uniparental stem cells, and we discussed about creation of them and some developmental potential and so on. We also learned about pronuclear transfer, right? And then what we now have to, there are ways of how there are protocols in the lesson. If you look at the one that is given, then you'll find some more details of protocols and other things. But otherwise, we have pretty good understanding or fair understanding of what we are actually, why we are doing this and how to do and so on. So the next thing is, say, for example, we have got the uniparental embryonic stem cells. And we need to then perform some experiments. And if you want to do that, one should require, say, I have two zygotes, and then we did pronuclear transfer, and then got one AG and one GG, right, by PNT. Then we are going to study some things or trying to check the developmental potential and so on. How can we be sure that what we have in our hands is uniparental? And if it is uniparental, how can we be sure that which one is AG and which one is GG, right? When we are mixing or trying to do pronuclear transfer, we cannot always be sure which one is male pronucleus or which one is female pronucleus. Even if we are sure, there should be some molecular characterization, definitively some proof that the one that we have here are AG and GG. So we need to characterize the cells before we perform further analysis, so that we are sure that what we are doing with is either AG, GG, or if it is not uniparental at all. For that characterization, there are several techniques. And one of them would include the analysis of chromosomes. And then we will also discuss, today's class is very important for this purpose. It will also show up in CIA exams. One of it will be about single nucleotide polymorphisms. And that is also involving PCR-based methods. So we are going to study, we are going to look at analytical techniques. I hope you can recollect a little bit about PCR from the last course. If not, we will have a quick review anyway. And in PCR, I would want you to also remember about primer designing, a little bit, or fundamentals of primer designing. Okay. So we will have a look at some of the methods where we can isolate the chromosomes and find it out. So analysis of chromosomes would be something like this, where we have to have a proper design of the experiment. It's not like after we get the zygotes and then we will analyze. It's not. Designing starts from where we get the zygotes from. We need to get two zygotes, right? And assume that we are going to, this is a zygote and this is another zygote. They are obtained from a parent, say, and one of it will be a mother and one of it will be a father, right? The same case here too. We will assume the same mother in both the cases. Assume that zygotes are made from the same mother and father. And we have a male pronucleus and we have a female pronucleus. They are biparental. And then we are going to perform the PNT, pronuclear transfer. So when we do that, we exchange the male pronucleus from one zygote to the other one. So our idea is eventually to get one AG that is androgenetic zygoid, and one GG gynogenetic zygoid, right? So, sorry, I made a mistake here. Let me correct it. So, yeah, if there are mistakes, please correct me. So this is these are two female pronuclei, so it is gynogenetic one. So if you want to molecularly characterize, we should have taken the mother and father. So mother A, the same mother here too. That's why I'm putting like that. Mother, father B, and father B. So we should have chosen some features here that are specific only to the mother and some only to the father. So that means they should be homozygous to something. Homozygous, I hope you remember, the opposite is heterozygous. Right? If somebody has gene capital A dominant and this is recessive, that is heterozygous. If they were having small a, or capital A, they are homozygous. The same copies on both. So if we identified a trait or a marker that we can work with or characterize, then it would be ideal if we choose the mother and father, right, that are homozygous for those traits. When we are doing, so in all the experiments, this is a general setup, right? And here, based on, if we have chosen a marker that has chromosome banding pattern, then we would look at how is the chromosome banding pattern in GG and, AG and GG. Then we can figure out if it is androgenetic or gynogenetic, or if they are not uniparental at all. So I hope you understood the general experimental setup, and I will try to go through this and see if we can understand. And then probably I will try to explain. Okay, I'll do it here. So we all know about chromosomes, right? And during metaphase, they will fold up like this. And this is the centromere, and we have the telomeres at the tips. If we add stains like GIMSA stain, which we will see the protocol in a while, if we have added GIMSA stain, some regions will get densely stained, and some regions don't get stained. So there is a kind of banding pattern for each chromosome. And sometimes there are chromosomal abnormalities where you can see it something like this. 1 second. So if we have a copy, 2 copies like this of, say, chromosome number 1, we'll call it as chromosome 1, and this is another chromosome 1. So in majority of the organisms, the chromosome size, the 1 is usually the largest one, and then second one will be the second largest, and so on, eventually the small ones. It's not hard and fast rule, but many times that is how chromosome numbering is done. Largest one first, and towards the end like that. So this individual that you're seeing here, for chromosome 1, the banding pattern alone, okay? We are not talking about specific genes or anything here. They are homozygous based on the banding pattern. If you look at, if I try to make it a different way, let's see if I can make it. So here, if you see, if I have an additional 1, right? There, we are talking about the same chromosome, chromosome number 1. Okay, but there is a difference. The individual here, assume this is the father, and the father or the mother, say, we will talk, this is the mother. Mother is homozygous in terms of chromosome banding pattern on chromosome 1. The father is also homozygous. There is a difference, right? The difference between them is this part. This extra band is there in the father, whereas it's not present in the mother. Now, during gametogenesis, what may have happened? What would happen is that if we see, I hope you notice the difference between these two, right? Keep it like that. We will come to this. So when the gametogenesis happens, they will separate out, or they will become what you call, they will segregate, and they'll go each to an individual. This will go to one individual, and this will go to the other individual. And the same thing will also happen with the father thing. One second, this will get a little bit. So what may happen here is that in the if we look at one set, one chromosome will come from the father, and the other one will come from the mother. And you see that the offspring, so the zygote that has formed now, is actually is heterozygous. These both are heterozygous. One has been obtained from the mother, and the other one from the father. From the mother, and this is from the father. They are heterozygous, and they are biparental because they got these chromosomes from two different sexes, or from father and mother. I should also bring a notice to it. What are these banding patterns? Some of them are densely stained. Those regions are called as heterochromatin. And there are very lightly stained regions. Those are called as euchromatin. So the difference is that they are all DNA, chromatin, everything. The staining is different in these ones because these are highly silenced parts of DNA, whereas euchromatin regions are highly expressed regions. Okay, that's why they have these staining differences. Anyway, getting back to what we were discussing. Now, if I am making uniparental stem cells, and I want to characterize, or if I was assume that we are performing pronuclear transfer right now, then when we do the pronuclear transfer, there are many things that will happen, isn't it? The whole chromosome set will move from one embryo to another embryo. So I'm trying to now make this pronuclear transfer. So I'm going to exchange this male pronucleus, although it is only represented by one chromosome, okay, chromosome 1. But I think you can understand that if it is happening with one chromosome, it would happen with the rest of the 23 chromosomes as well. As a part of male pronucleus, I am going to take this set from male, and I'm going to put it into another zygote, the parental zygote. I am going to take the female pronucleus, and I'm going to put it here. And they are in two different zygotes now, right? Because I performed pronuclear transfer, you can also expect that something like this would have happened. And now if you see that both are from female, how can I say that? I'm looking at the banding pattern. Okay. The same here it is. This is gynogenetic, and this one is androgenetic ones. Okay. So that's a general or simple principle of way of doing things. So now we will look at how the method is usually done, general protocol. And I hope you will be able to see and understand that. Okay, sorry. So what they do for a karyotyping, typical one, okay? Here, usually they do use this method to find out many different types of chromosomal aberrations and so on, any chromosomal deformities, inversions, deletions, insertions, they can find out using this technique. This is little primitive, but it is still popular. So normally they would have taken cells from the blood, and they would add phytohemagglutinin and other things and incubate it at 37 degrees. They will culture them for 3 days, and then they would proliferate the cells that can be divided, not the red blood cells, but the other ones. And then they would add colchicine to kind of inhibit any further cell division. So those that have come into metaphase will remain as such. And they would put it in hypertonic solution. What do you mean by hypertonic solution? If you take cells and put it in 3 different, this is a classic experiment with red blood cells if you have seen in any textbooks. If you put it in isotonic, iso means this is the cell, the concentration outside and inside the cell of the salts or ions is equal. So you call it as isotonic. If these cells are here and you say hypertonic, sorry, HYPER, hypertonic, that means the salt concentration here is very high compared to what is inside. So what happens? Water moves out and the cell will shrink. Here we're putting in hypotonic, meaning that where the salt concentration is less, and therefore water from the outside, it is relatively high in the cell. So the water from the outside will enter into the cell. As a result, the cell will bulge like this. Okay. So what we made is we took dividing cells, we kind of fixed some of the cells that are about to divide in metaphase, and we pumped water into it by putting it in hypotonic saline, and then we make a splat. We drop one droplet of that solution containing the cells onto a glass slide. When it hits, the cells will burst and they will splatter, and the chromosomes that were in one cell will fall and spread out on the glass slide like this. Then we fix it, we add trypsin to digest some of the chromatin and other proteins, and then we add GIMSA or other kinds of stains are also there, GIMSA stain, and then we wash it off. And then on these chromosomes, we will have some regions highly stained and some regions poorly stained. And once that is made, they arrange them. Usually they would have taken a picture of it, cut each of the pictures, and they would arrange. They will make pairs of chromosome 1, 2, 3, and so on. So the same thing can also, if you look at the picture, how it would look like is something like this, where the chromosomes, these are the banding patterns, some are highly stained, some are poorly stained, and we can look at them and figure out if there are any abnormalities. For our purpose, we would have been trying to look at if the banding pattern on these 2 chromosomes are homozygous or not. And which chromosome are we looking, and which part of the chromosome we are looking? We should have had the prior knowledge about what we are actually looking for. Okay, so if we designed our experiment properly, and then we could look at those particular data and figure out if that particular band is present or not. That is how karyotyping is used for characterizing uniparental stem cells as well. So this is how nowadays, of course, there are computers and computers with the powerful microscopes. They can take a picture. When they take a picture, it would look like this, and the computer can pair them up with I mean, image analysis and so on. They can pair them up, and they can also give us banding patterns like this and so on. So it makes it much easier. But what we are going to discuss now is something that is of interest to us. Before going there, I will quickly go through the PCR, and then we will probably end up there. I hope we are now learning something so that we can use PCR to characterize, and this is likely to come for CAA, and we will discuss about the date and other things later. So if you remember a DNA molecule, 5' and 3', and assume I'm trying to, okay, let me put it here itself. And this is the second or complementary strand, GTCT. This would be the 5' to 3' end. So if I want to design primers, and I design primers like this. So primer is a small stretch of nucleotide. Say I would like this. It will be, this would have been the 5'. Is that right? No. One second. I wanted to make the mistake first. So if I took a primer TC, if I designed a primer like this, and also CAGA, then this will be the 5'. No, this is the 3'. This will be the 5'. And this is 5', and this is 3'. If I add DNA polymerase, then DNA polymerase, you have to remember, polymerization always occurs from 5' to 3', right? 3' OH is important. If I have designed primers the way I'm showing right now and add DNA polymerase, then the polymerization would have occurred in this way. So they are not very useful. This is actually, these primers are wrong. So we should always have the primer that will converge, right? Instead, in the primers that I've written, they would have polymerized. If there is DNA, they would have polymerized in the opposite directions, and that's not useful to us. Instead, we should have, one second. So we should have designed primers in such a way that they would have been this way. A, G, A, G. This is 5', and here is the other primer, something like this, 5', and this is 3'. Now, if I add DNTPs, DNA polymerase and all, polymerization would have occurred in this direction. And on the top strand, it would have occurred in this direction. And eventually, we would have gotten two copies from one template. And these two copies would serve as the template for the next round. Then you'll get 4, then 8, and so exponentially, they will keep on increasing. That's the general concept of PCR. Today, what we are going to use it for is characterizing. As I described earlier, we should have had a plan for the experiment. We should have known the molecular structure of the experiment itself. So when I started this, I have described general scheme. We should have had an idea about what we want to do, which molecular marker we are looking at. We should have had the proper plan so that we can choose homozygous parents, so that we can keep track of the zygotes, pronuclear transfer, and then be able to characterize the uniparental stem cells. So the same thing is here. Here in the PCR-based one, there is a gene called GPI1. We are not bothered about what does it do and so on, but we are interested to use it as a molecular marker. There are two alleles called, say, allele A and allele B for GPI1. GPI1 has two alleles, allele A and allele B. Allele A has about 100 amino acids, which will give a protein of 100 amino acids. Allele B would give a protein of 150 amino acids. I guess you can calculate the size of the DNA, isn't it? This would be approximately 300 base pairs, and this would have been somewhere around 450 base pairs, right? So that means we should have started something with, we should have taken the mother and the father who are homozygous for like this, small a, and the father for small b. You can also take it vice versa. The point is to have two different alleles like GPI1AA, homozygous for A allele and homozygous for B allele. Then the crossing would have happened, or in vitro fertilization, assume that, and we would have gotten two zygotes. One of it has an allele A and one allele from the father B for the GPI1. In the other zygote too, we would have it this way. So they are heterozygous and they are biparental. Now what we should do is pronuclear transfer, PNT. And if we did PNT, if PNT was successful the way we intended it to be, then we should have gotten something like this, homozygous, uniparental, right? So when if we can only ascertain that they are homozygous by using some of the techniques. One technique that by chromosomal banding pattern we have seen karyotype, and the second one that we are seeing is using PCR-based method. We will also see one more method after this one. Okay. So for this is the general experimental setup. Now we will just see how it works, and then we'll go ahead. So this is where PCR primer is there, and I also want to give you another insight into general principles. Say, for example, there is a DNA like GPI1 we were talking about, and this is the A allele, and it is about 300 base pairs, right? And B allele was about 450 base pairs. And this is larger in size because there was an insertion in it. There is an insertion of about some 150 bases, say. And that's why it is larger. Just assume that to be so. Now I want to design primers so that I can do PCR on these two. Sorry, 5 prime, 3 prime, 5 and 3 prime. So if we designed primers, assume the primers were made like this. One of it is this way, and we call it as primer F, forward, and we call it the other one primer R. The same primers can also be used for the allele B as well. Forward and reverse primers. If an individual is homozygous for A, okay, homozygous, and we perform PCR with using forward primer and reverse primer, the size of the product would be approximately 300 base pairs. If an individual is homozygous for BB, and we perform PCR and other things as well, the product size should be around 450 base pairs. If somebody is heterozygous and we perform the PCR, what would it be? They will get 2 bands. one band from allele A and another band from allele B. So they would end up getting a 300K base pair fragment and a 450 base pair fragment in the electrophoresis. Okay, I hope you understood this one part, and now we are going to repeat so that you will see it in a scheme, how it look like in gel electrophoresis. So let's see how we do it. We should become familiar with analyzing the data, interpreting from the data, or being able to draw a data how based on the end result if somebody has given us. So I'm writing about, I have taken the DNA from some cells. I have taken from sample 1, I'll call it as sample 1, sample 2, and sample 3. I've collected DNA from each of them, and I performed PCR using forward and reverse primers, and then I have done electrophoresis. I hope you remember electrophoresis. That is mobility of DNA in an electric field in agarose gel, and the smaller ones will run faster and the larger ones run slower. This is the ladder one. So I will put as 200, 300, 400, 500, and 600. These are the bands of the ladder. So if we have done PCR, and I'm going to show you the results, and then you can probably predict what it means. So A has one band here, and B has one band and corresponding to 300, and another one here, and 3 has something like this. So based on the gel that have drawn a schematic of the gel, what would you expect? What do you think is A? Is it heterozygous or homozygous, and for which allele? Remember, we are talking about GPI1 allele, and there are 2 alleles, A and B. A is about 300 base pairs, and B is about 450 base pairs. The actual details may change, but the principle is what we are interested in, the ability to interpret the data. Anybody? What do you think A is? Sample 1 is?
Homozygous AAS.
That's right. If it were, there is no B allele at all. If it were there, we would have got another band also, right? Sample 2?
Heterozygous AB.
That's right. This is A and B. And 3?
B, sir. Homozygous.
Homozygous for B and B. So can you tell, if anybody can tell me which of these are uniparental based on our previous experimental setup? I'll just show you the previous experimental setup. What did we discuss? Not here. Okay. This is the experimental setup, right? We've taken a mother who is homozygous for A, father homozygous for B, and we made these zygotes, and then we performed pronuclear transfer, and we got some things, and these are 1, 2, 3. Assume we made 3 or so. Then based on that experimental setup, what can we say? Which ones are uniparental and which ones are biparental?
So 1 and 3 are uniparental, whereas 2 is biparental.
That's right. This is biparental, and we do not want this as per our lesson we're speaking. This is uniparental, and this is uniparental as well. And which one is homozygous? Which one is AG and which one is GG? I'll show you again. You can see from here and try to predict which one is androgenetic one, which one is gynogenetic one. Okay, you saw this?
One is gynogenetics.
So
One is, yes.
androgenetic.
Let's look at the data again, so that look again and both of all, and then get to the answer. A is sorry, sample one is
Gynogenics.
gynogenetic one, and B is androgenetic one. Okay, you can only interpret these properly and correctly if we had the knowledge of the experimental setup. That's why I have taken time to describe the experimental setup. Okay, so there is, I hope you remember in our previous course we learned about point mutations. In point mutations, we also learned about single nucleotide polymorphisms. Single nucleotide polymorphisms, or you call them as SNP. What does it mean is that you have, if there is a sequence, say for example, I'll just write the gene GATC A, and there is a DNA GACT. No, I should not write the same. So DACGACCA. So the bottom strand will also be something like this. Right? Okay. And this is the 5' end, this is 3', and here is, sorry, 3' end, and this is the 5' end. So if there is another DNA, almost same thing, okay? And if the DNA is something like this, except there is one difference though. The difference is, say, for example, here. So what is the difference between these two alleles? I will give also some names to these alleles so that it will be easy for understanding. I'll say allele allele P, P1 and P2. Okay. So what is the difference between these two alleles? Is this region. Right? In that position, we have a difference. That is, one is having A in this position, whereas the other one is having G. The rest of the sequence is same. We cannot, like in the previous one, we cannot use based on size. In this case, we used size as a molecular marker. We cannot use the size here as molecular marker because they both are, the length of the gene or fragment, whatever, is same. The only difference, however, is this SNP. We can still use PCR to find out whether a person is homozygous or heterozygous. So I will try to give you an introduction today on this, and if required, I will repeat it in the next class as well. But you should understand it well. It's a lot of fun because it is like a puzzle if you are trying to look at it properly. So I want to design primers for both of these. So I will design GATC A, right? That is the correct primer. And I want to also design primers here too, like this, right? Then they will polymerize in this direction. So the primers are good. I can also, the same primer can also be used here. And here, GATC, I cannot write A here. There should be something else, and that is G. Right? There is a difference. So there are three different primer sequences that we are using. We'll call this as primer 1. Okay. We'll call this as primer 2, and we call this as primer 3, and we call this as primer 2. Why did they call it 2? They are same sequences. Whereas here, primer 1 and 2, 1 and 3 are different. How much different? It's just one base, but it is still, they are different sequences, so they are different. Okay, try to have a mental map of this, and maybe we will try to also make a picture out of it and then see if we can make that happen. Okay, there are other things. Yes, I'll try to take this, okay, so that you will probably remember. So here is the experimental setup that we have discussed. Okay, so I'm putting it here so that it will be a good quick reference. Now think of it like this. I have taken a primer. I'll go to the previous one so that maybe. So here I'm going to do the experimental setup again, so that you will know what it is, what we are actually doing. We take two individuals, mother and father, and we should have chosen them to be homozygous or homozygous in either case. We discussed about two different alleles. We discussed about P1 allele and P2 allele. So we should have taken male to be homozygous for P1, and mother with P1, and father homozygous for P2 allele. Then if we made zygotes from them, say two of them, so they would have been biparental, so they would have inherited P1 and P2 each, and they are heterozygous. And then we would have performed pronuclear transfer, and if it was successful, they should have been P1 and P2 and P2. Okay, so I'll also try to take this and then let's see if we can take this up. Let's check. Yes, this is here. This is also the part of experimental setup, so that you'll remember. And now I'm going to directly assume that I have isolated the DNA from these cells. I have isolated DNA from these cells, and then I performed PCR, and I have gotten some results. And I want to see if you all can, if you can figure the answer out. And for each sample, we are performing two reactions. So how many do we need? Okay, we'll put 3. Okay, we have gotten 3 different samples. Okay, in this one, we have taken, say, sample 1, 2, and 3. And that is what we are going to use here. Sample 1, 2, and 3. Okay, this can get confusing. I will just, I will change a little bit the terminology because we are using 1, 2, 3. We will call them as samples A, B, C. A, B, and C. So here is the sample from A. This is from sample B. This is from sample C. In this one, at this point, I have done PCR with primer 1 plus. With primer 1, you can see here, primer 1 and primer 2. In the second reaction, I have done PCR with primer 3 plus 2. The same way for all the samples. Okay, so the size of this DNA, if there is, it would have been 300 base pair product. So now I'm going to tell you how it would look like, and you should tell me what is the genotype of the ABC. So for A, both these two are A, okay? These two are for B, and these two are for C. Sample of DNA that we have taken. So we get something like this. What does it mean? If you can remember in the previous time when we have learned about PCR, if these two do not base pair properly, okay, if I add the same primer that is here, say for example, if I put this primer in the bottom one, what would happen? If I add this primer, will it give a product or not? It will not bind, right? It will not bind and it will not give a product. Now look at this sample and try to figure out. And if you don't get the answer, I will discuss this in the next class. I will wait for you to finish, or try. Anybody finds out and tells me the answer for it. Please try. If you have any questions on this, you should, or doubts, then you're free to ask as well. One second. I have 6 people. Yes, please. I think you can solve. You are allowed to. You can please turn on your mic and you can ask questions so your friends can answer, and collectively we can get to the answer. Anybody? What does it mean? If you're trying, I will wait. Please tell me if you're trying or not. If you're not trying, I can continue probably.
Sir, A is actually biparental and heterozygous, sir.
Why?
It has both the primers, 1+2 and 3+2.
Yes. So why did it give? I'm just answering so that everybody will understand how you arrived at the answer.
So it has both the chromosomes. On replication, it gives.
I will say both
are
alleles,
equal.
right? Yeah.
Sorry, sir, alleles.
Yeah. That's right. So if there is a product here, that means what allele is present? It would mean P1 is present. That's right. If there is a product here with primer 3 and 2, what would that mean?
P2 is present.
That is P2. So this is heterozygous. What about B?
B is uniparental. It just has P1 allele.
P1, and there is no band here which says there is no P2. So it must be P1 also, right? So it is homozygous for P1 allele. What about C?
C is a P2 allele, sir.
Yes, it is homozygous for P2 allele, and there is no P1. Right? Very good. Thank you. And so I would also want you to tell me which one is AG, GG, and other things.
So P1 is GG.
No. You have to speak in sample A is.
Sorry, sir. Sample A is biparental, sir.
This is biparental.
sample B is GG and sample C is AG.
Yes, sample B is gynogenetic and sample C is androgenetic. Okay, these are some of the methods that can be used to find out which of them are, whether they are uniparental or not. In science, we always want to be in touch with, we want to be certain of what is happening. And only then experiments are actually experiments. Otherwise, if you are leading to ambiguity or we are uncertain, maybe then it is not conclusive. So before performing, say, for example, chimera, before doing anything like camera technology and all, it is better to verify the samples before proceeding to further complicated experiments than just going ahead and not doing much. I also want to, I think we have 3 minutes. I just want to have quick questions on CAA exam. Whatever will be finished until Saturday will be in your syllabus. Okay? So in your syllabus for CAA exam. So please keep that in mind. That is one thing. Second thing is, I want to know if did we have a discussion on the ethics? Yes or no?